Hello everyone, my name is Samuel Diebolt and I'm a PhD student working with the Physics for Medicine laboratory in Paris, along with the Institute of Psychiatry and Neuroscience, also in Paris, and the Iconeus company. So functional ultrasound imaging is becoming an increasingly important tool for the study of functional connectivity in awake animals. And in recent years, a huge amount of data was acquired from longitudinal pharmacological studies using functional ultrasound, and thus offering us insights into the brain functional connectivity without the heavy biases induced by anesthesia that we could have with fMRI, for example. However, fUS imaging still has significant challenges, and in particular data preprocessing that still presents significant challenges, in particular considering the confounding effects of artifacts caused by animal motion. So during the last 6 months, I have been working on benchmarking several pre-processing strategies and their respective performance. And these strategies were previously used in functional connectivity studies using functional ultrasound imaging, along with new methods inspired by fMRI and fMRI data pre-processing pipelines. So while this study is still preliminary, we've been able to find several axes for improvement to the pre-processing of awake functional ultrasound data. So first, we've showed that masking the IQ data before clutter filtering allows better separation of the tissue signal from the blood signal by the SVD-based clutter filters. And indeed, this is due to the fact that regions outside the brain, such as these, can contain areas of high energy signals that can easily bias the singular value decomposition. Second, we implemented a new adaptive singular value thresholding method that better estimates the size of the tissue space respective to the blood space during the clutter filtering. And this new method allows better separation and better estimation of the tissue space and blood space, and also allows better removal of the motion artifacts even before the power Doppler computation. And finally, in the rest of this study, we discuss the effects of several data cleaning methods, namely scrubbing, standardization, and nuisance regression. So we first leveraged the motion tracking data offered by the Neurotown mobile homepage to select and remove problematic frames that were affected by motion artifacts. Then we adapted the nuisance regressors developed for fMRI data preprocessing that go beyond the simple global signal that is usually used in today's fUS literature. So with all these new steps, preliminary results showed that the best pipeline was able to entirely remove background artifacts on seed-based statistical analysis, thus resulting in clearly defined functional fingerprints. So in this study, we identified important axes of improvement for the pre-processing of functional ultrasound data, and we are convinced that we will be able to find a reproducible pipeline that will be robust to motion artifacts.